The overall goal of this procedure is to clone and express antibodies produced by B cells. This is accomplished by first isolating B cells from a relevant source tissue, like blood or thymus. The second step is to perform FACS and select the B cell population positive for IgG production. Next, the cells are immortalized by infection with Epstein-Barr virus together with toll-like receptor 9 activation by CpG-ODN 2006 and kept in culture. The final steps are to isolate RNA from clones that are tested positive for IgG production by ELISA and to generate copy DNA from both light and heavy IgG chains. Ultimately, the IgG sequences can be cloned in mammalian expression vectors to produce high quantities of monoclonal human IgG.
The main advantage of this technique over existing methods, like phage display technology, is that original IgG heavy and light chain pairing is not lost.
This method can answer key questions in the field of autoimmunity, such as discovery and functional characterization of pathogenic autoantibodies.
Don't forget that working with patient material and EBV virus can be risky. EBV virus is a human pathogen, and precautions such as BSL-2 procedures and wearing adequate protection should always be taken while performing these steps.
Before staining the PBMCs, plate them in a 25 cm flask with 6 ml of complete rPMI 1640 medium and let them recover.